This is a video about antibodies by Matt Mackey, Todd Spigner, and Megan Evans. So, antibodies are proteins found in the bloodstream, and they're large, Y-shaped molecules, all of them of which are Y-shaped. They are also called immunoglobulins, and they contain 4 polypeptides exactly. They specifically are binding proteins, much like other proteins that bind to ligands. These bind to very specific ligands in the bloodstream, which are known as antigens, or foreign substances found within the body. Where antibodies are found, like I said, they're found in the bloodstream, but they can also pass through capillaries, and sometimes they may leak into the body's extracellular fluid.
So, why are antibodies necessary? Well, antibodies identify foreign substances that are in the body, and they also help the body neutralize these substances that it finds. And it can do it in 3 different methods. It could bind directly to the surface of the antigen and kind of coat, and it'll stop, or so you can say, slow down the antigen from doing its job. It could also stimulate the body's other immune responses, such as phagocytosis or phagocytes. Or the last one, it can mark the pathogens so it can be identified and destroyed by other immune cells such as white blood cells and macrophages.
So we're going to talk about the case when an antibody binds to an antigen. First, I will draw a diagram of what is occurring. Here's an antigen. Okay, so each antigen typically has several different receptor points, which are also called epitopes, and this lymphocytes can bind to these sites, these epitopes, and each trigger a certain reaction. In this case, when a B lymphocyte or a B cell, or a T lymphocyte or a T cell binds to either any of these sites, a response is triggered to where an antibody is released, and then what occurs is the Y-shaped antibody, which kind of looks like that, is applied here with like the same receptor shape. And this is just like a stick figure drawing, which is also done in normal science, and it can have different types of receptors as well. But here is just the general thing where that's not even a Y. But here is just the general thing where the antibodies bind to the epitope sites on the outside of the antigen.
Here we're going to talk about stimulate and mark at the same time because their functions go hand in hand. This is the most important method by which antibodies work because once the antibodies have bound to their antigen, they undergo a slight change. So, like we were talking about before with the antibodies binding, this conformational change enables the antibodies to activate other proteins called complement proteins. And pretty much what these complement proteins do is they highlight this antigen for other cells to come kill it. If it doesn't do that, the complement protein could also cause a burst within the antigen cell, just instantly killing it. So it just explodes. And yeah.
This is a graph which represents how the first exposure of an antigen in the body and the second exposure of an antigen to the body is represented. So the left side, the y-axis, represents the concentration of antibodies within the body, and the x-axis represents the time in increments of 7 days or a week. So where the first exposure would occur would be right at 0, where the first exposure is And notice how there's no visible line here. That is because this is the time it takes for the body to recognize that there is a foreign invader within your body, an antigen. And around a week's time, the body is able to produce antibodies against the antigen. And this, being the first exposure, is the time where you usually display signs of being sick and have symptoms of the specific virus or thing that is affecting you or afflicting you. So in the case of measles, you get the first exposure, you get sick here, but you're also producing the antibodies to fight it. And so when you're not sick anymore, you gain a certain level of immunity to where the second time, if you're so unlucky, you get exposed to measles once again a couple weeks after your first exposure. What would happen is that your body, your memory B cells, would be able to come in contact with the antigen. They would recognize the antigen as one that you've seen before and produce the antibody necessary very quickly, much quicker than your first exposure. So at this point, you typically would not get any symptoms of being sick or any signs of having the measles again.
Arr, let me tell you a drunken sailor tale. There once was a young girl named Jocelyn Brown, and her mother had looked at the world with a frown. There was too much disease for the eyes to see, so she said, "Measles, measles everywhere." And J. Brown said, "Hey, why should I care?" And then J. Brown went to a measles party against her will. Poor J. Brown, not knowing, contracted the measles, but Mother Brown knew far better than she, for antibodies made by B cells will know that the measles at that party is conquerable. So the next time J. Brown contracts the measles, she practically would not even know, for memory B cells secrete antibodies remembered from that one measles party. Arr!